"350 years ago, there was a Belgian scientist called Van Helmont who carried out an experiment that I'd like to tell you about. He was terribly interested about the origins of life and how life started. And like a lot of people at that time, he thought that life arose spontaneously all the time. It was just springing up from nothing all the time. And in fact, people like Aristotle and famous Isaac Newton thought this. Well, Van Helmont's experiment was the following. He said, I took a box and I put into it a dirty undergarment and some wheat. And I waited 2 weeks. And what do you think he found? Let's see. Would you like to see what he found there? We'll take the lid off.
What did he find? Oh,
what is it?
It's a little mouse. No, where is it?
Can't you catch him? He's alive anyway, isn't it?
He's alive.
Okay, let's have a look. This is what Van Helmont found. And he found it in this box, which contained wheat and dirty undergarments. And so he concluded that life had arisen spontaneously in this way, which was a silly thing to do, wasn't it? Do you believe that?
Yeah.
Well, you know I put it there first, but if you had found it yourself under those conditions, you would have nowadays said it crawled into the box. But that wasn't so obvious in those days. There were other examples of this sort of thing. Everybody knows that when you leave a piece of meat out, maggots appear spontaneously. Here's a piece of meat, and you see it's crawling with little animals called maggots. What are maggots? Yes?
Larvae of flies.
Larvae of flies, rather like caterpillars from butterflies, aren't they? And they arose spontaneously, didn't they? They just arose from nothing. No. Where did they come from?
Flies.
Flies, yes. There was an Italian called Redi who did the good experiment. He took another piece of meat, put the two alongside each other, and covered this one with muslin, and he found no maggots. He found the flies trying madly to get in at the meat and laying their eggs on the muslin, but they didn't get in. So, all right, we can understand how that arises, but when the microscope was discovered, things became apparently even more difficult, because under the microscope there was a whole new teeming mass of life. Let's just have a look at a drop of pond water under this microscope if we can. And you see, I hope, under that microscope, you see a mass of little animals so small that you can't see them, but millions and millions and millions of them in a drop of water. And here it was really difficult to believe, Lightspeed, it was really difficult to believe that these things could possibly be reproducing and coming from other little animals. In fact, where did the little animals come from anyway? They come from the air. The air around us is full of bugs. You're breathing in bugs all the time. There are millions and millions of them in this theatre, all the time on every little speck of dust. And that's how they arise. If you take some soup, or this is turnip juice, as a matter of fact, and you can boil it and kill everything in it, but then you just leave it, it's open at the top, you see, you get this sort of thing happening, a mold forming, which is a living thing. And if you look at this stuff under the microscope, you'll find it swarming with living things, just like the specimen we've seen under the microscope now. Well, now, where do those arise from? People said they're in the air. And this was proved by Spallanzani and others by boiling this, taking 2 flasks, boiling one, and sealing it off. And it remained clear as Amontillado sherry. And so they said, well, that's fine. You see, the bugs can't get in, and so they are coming from outside. But then some difficult people said, yes, but that's not fair, because obviously bugs like you and me have to breathe, and they want air, and you've stopped them having any air, so they die. And it was Louis Pasteur, a brilliant scientist in many ways, who did a very clever experiment. How could you do an experiment which would allow the bugs to have air, but not allow the bugs in the air to come in? Well, you have to have something which lets through air and not the little animals floating around in the air. And this is what he did. He used what he called a swan-necked flask, made this shape, and this is open at the end. You boil it, it kills everything in the flask. Then as it cools, the air comes in, but the little animals are caught on the side. And this was made by Elizabeth here. This was made over 2 months ago, and that also is as clear as a bell. And so the idea of spontaneous generation seemed to be destroyed. Pasteur had done that experiment just about 100 years ago because the French Academy offered a prize for somebody. They were tired of the argument. They wanted it settled. And they said, we will give a really good prize to somebody who can settle it once and for all. Pasteur did that experiment, and he said, never will the theory of spontaneous generation arise from this mortal blow. So there we are. We don't believe anymore that life is arising all the time. And we have to ask, how does it arise? What makes living things? It must have arisen a long, long time ago if it's not arising today. Well, you know from what we said in the last lecture that life is a very organized, ordered affair, and it couldn't easily arise spontaneously by any method we know, but what is it outside the Earth which makes it possible? Sunbeams. The light of the sun makes this all possible, and what I want to do now is to try to trace the natural history of the sunbeam through the growth of life from the beginning. And the first question we must ask is, how long has life been here on the Earth? And before we ask that question, it would be sensible to know how long the Earth has been here. Well, how can we tell how long the Earth has been here? Yes?
Well, by rocks.
By rocks. There's a clue here, isn't there? Yes, by rocks. We can have a look at rocks. We know how old the rocks are by using radioactive methods, seeing how fast the elements in the rocks, how much of the elements in the rocks have decayed radioactively. And then we look at the rocks, and here's one. This one is 450 million years old. It's from this country, and it's from the bottom of a seabed, and you can see on it some very, very clear formations of shells. So what do we know? Shells must have existed 450 million years ago. And we can go back a little further than that, but not much further. As we go back to about 700 million years, the fossils disappear, although rocks are much older. Here's a bit of Scotland, the Western Isles of Scotland, and this is 2.5 million years old, but there are no fossils in it. So I've got a time clock here which may help us. Let's just have a look at the age of the Earth and life. The clock starts at midnight here, and we'll go backwards in time in billions of years, 1 billion, 2 billion, 3 billion, 4 billion years. Now, I have just shown you a bit of Scotland, which I said was 2.5 billion years old. Now, billion, when I say a billion, I'm going to talk about the American billion, 1,000 million. It's smaller than the English billion. I suppose the English should be proud of their big billion, but the American billion of 1,000 million is more convenient, so we will use that. And I've just shown you this rock then at 2.5 billion years, and the oldest rocks that are known on Earth are 4 billion years old. These have only recently been discovered, and there is one of them in the Geological Museum who kindly lent these to me at 4 billion years. But there is other evidence that the crust of the Earth can be dated at 4.5 billion, that's 4,500 million years old. Better still, the meteorites which come from outside the Earth and which were, everybody believes, created at the same time, are 4.55 billion years old. And the Apollo spacecraft, not 11, but the later ones, brought back rocks which were 4.6 billion years old. So everything is round about there. 3 or 4 independent measurements tell us that the Earth is 4.6 billion years old, and that's something we can be fairly happy and certain about. Now, when it comes to how old is life, things are not quite so certain. But if we go right now, this is a clock on which I put hours as well, and it may help you to think of the hours as the hour on this clock would be 400 million years, wouldn't it? If that's 4.8 anyway, it would be 400 million years. Now, as I've said, the first fossils start about 700 million years ago, and they're soft animals, and of course they don't make good fossils. Unless you have a good hard backbone or a shell, you don't make a very good fossil. Not that anybody really wants to make a good fossil, but that's the geologists wanted. And we saw the shells at about 1.5 hours ago. Fish appear a little later, and all this is in the sea, and there is nothing has crawled onto the land at all until 1 hour before midnight. That's 400 million years ago. Then we begin to get the land plants and the reptiles, and some rather big reptiles which came about there, which you may have heard about, the dinosaurs and so on. The mammals only appeared about 20 minutes ago, and the mammals and the birds, that's on this scale, you know, about 150 million years ago. The warm-blooded animals, birds and the mammals. Primates, a bit like us, the monkeys and the apes, appeared 1 minute ago, and still no man. And man appeared 10 seconds before the clock struck midnight. So we haven't been around very long. Man is only a very, very small part of the history of life, and even more so of the Earth. So now we want to ask what happened before that. And when I said the oldest fossil was about 700 million years, these are big fossils. But people are very interested now in doing research on the origins of life. And they are interested in much simpler, smaller animals, because presumably the first animals would be very small, wouldn't they? And so you look under a microscope at rocks. And if you do so, you find more fossils, perhaps not so nice as the shells and things we saw, but you find fossils like this. Now, this is a fossil which was found in Gunflint rock formation in Canada. And that rock formation is 2 billion years old. And this is a little plant. It's an alga. It's very similar to some which are still around today. And so we know for sure that there was life 2 billion years ago. That is way back here. And if you look at some of the oldest rocks, the rocks particularly in South Africa, the fig tree formation. The gunflint, these are funny names. The gunflint is called the gunflint because they made gunflints, flintlock rifles from it. The fig tree formation because there's a place called Fig Tree quite near to it. And the fig tree formation is 3.1 billion years old, and in that you find little fossils which I won't show you pictures of because you wouldn't believe me, and I'm not sure I believe them myself. But the experts on these do. There are little fossils which are, everybody believes who studies these things, fossils of bacteria. And possibly fossils going back further because there aren't many rocks. You see, the rocks are melted and destroyed by erosion and so on. So there aren't a lot of old rocks around. But on the whole, it's round about here that what we might call life will have started. I've put it as a grey area. The black is probably dead. The white and onwards is certainly alive, and this is a grey area, and this is about where life started. Well, now we have to ask how that could have happened. All right, we have the sunshine, we have the sunbeams, we have our Earth with its atmosphere and the sun streaming in onto the Earth. We have some molecules around. How could they make life? Well, to answer this, we must first of all ask, what is life and what are living things made of? What are the building bricks of us? What are we made of? Well, around me you will see some molecules. And those of you who are not chemists may be eyeing these with some sense of resignation and thinking, I'm not going to understand this, because they are terribly big molecules. But it's easy. It really is easy to understand most of the things that you should understand, we need to understand for this lecture. Nearly all these living things are made up of 4 kinds of molecule. 95% are made up of carbon, nitrogen, oxygen, and hydrogen. And if you can remember black, blue, red, and white for these, it will help because that's the code we use. Now, one other thing I'd like you to know is that this carbon has 4 prongs on it and forms 4 bonds, so that if it links with hydrogen, it does so to form 4 hydrogens, CH4, and do you know what that molecule is, anybody? Methane. That's North Sea gas. And ammonia makes 3, and nitrogen makes 3, and that's ammonia. Now, I know none of you will know what this is. Oxygen forms 2 bonds. That's H2O, and hydrogen forms 1. Now, if those are the simplest molecules you can make, and you just treat those as building bricks, you treat them as you would treat building bricks from which you make anything, and almost anything you can make from those will exist chemically. If you just break them up like this and put them together again, if you can put them together and there are no spare bonds, it exists. Does anybody know what that one is? Very good, methyl alcohol. And here's ethyl alcohol. I shall switch scales as we get to bigger molecules because otherwise we should fill the room. That's ethyl alcohol. And now let's have a look at the things we're all made of. What are little girls made of? Sugar and spice and all things nice. Well, I've got some sugar and spice out here, I hope. Have we? Yes. Let's hold up our molecules here. What are you?
Glucose.
Glucose. That's a sugar. That's barley sugar. And what are you? Fructose. Hold it up so we can see it. Now, if those 2 are linked together, we get ordinary cane sugar that we put on the table. What are you? Sucrose, which is cane sugar. And now if we link a lot of those together, we get, hold it up, cellulose, which is longer than that. I couldn't make it any longer because she hasn't got a very long arm. But that's cellulose, which makes the cell walls and cotton and so on. What's that? Starch, which is very similar, and that feeds the plants. So those are the sugar and spice and everything nice that the little girl's made of. But, and one other molecule I'd like to show you. Can you hold it up by that end? Because that's the way it likes to be. This is a fat. You're a fat, okay? This is called lecithin, and it has these oily tails. We shall come back to all those in later lectures. But what I want to come on to is the two most important molecules which really life has to have. However simple life is, it has to have two molecules, and those are DNA, deoxyribose nucleic acid, but we'll call it DNA, and proteins. And it's almost impossible to imagine how life could exist without these two, because the DNA is the programmer. It's the message. It tells the organism whether it's a man or a mouse, whether it's a boy or a girl, even whether it's a leaf or an elephant. The only difference is that, and DNA, I've got a different model of DNA here because it's a little bit difficult to see in that model. Here I've left the atoms out, you just see the bonds, and DNA, you see, is made up of just 4 molecules called the nucleotides. I won't bother you with their names, but all that is just made up of 4 types of molecules called the nucleotides. And it's just the order of those nucleotides in this long DNA molecule which says that you, what's your name? Yes. Simon. Well, you're Simon because of the order of these things in the DNA. There may be another Simon, but he'd have a different order of things. Now that programs the proteins. And here is a protein. It's one that we're rather proud of here because it's the first protein to have its structure determined. It's called lysozyme. It occurs in tears and in egg white. And the structure of this was determined by Dr. David Phillips here about 10 years ago. Now, that looks awfully complicated, but it's made up of amino acids. And there are 20 amino acids, and there they are on the Christmas tree. I've hung them all up on the Christmas tree, and you can have a look at them afterwards. This says threonine. This says Happy Christmas. But it also says aspartic acid, methionine, and so forth. There are 20 of those, and they are linked together. This is on a smaller scale, you see. I have some smaller scale ones here. This is 1 amino acid. That's 2. That's 10. Thank you. We will put a bit of string around it so we don't lose the end. There we are. That's 10, 20, 30. This is lysozyme. This lysozyme is all coiled up, and it's this long chain coiled up. 40, 50, 60, 70, 80, 90, 100, 110, 120, 129. There are 129 amino acids in that long chain, and not only are they 20 amino acids precisely that I've told you, but the order is absolutely precise. This amino acid has to be before this one, and that's what determines the way they're coiled up like that. So, insofar as the enzymes and the proteins and the DNA make up life, build our bodies, the enzymes do all the work, insofar as that is life, what we have to start with is to ask, how did these 20 amino acids and these 4 nucleotides, 24 molecules, will determine everything, from the simplest bacterium to the biggest elephant. They're all determined by the order of these things. How could they arise? Well, they arose, as I said, by the sun on the atmosphere. But the trouble is, it's a chicken and egg problem, you see, because these organic molecules all arose from living things. Of course, chemists can synthesize them, but most chemists are alive. And therefore, it's a chicken and egg. They arise from living things, and yet living things couldn't start without these organic molecules, could they? And the trouble is, in our atmosphere as it is today, with so much oxygen around, living things aren't very stable. They're rapidly consumed. For example, here are some living things which I will burn. It's rather cruel, but it's only pollen. It's living pollen. And if that is heated as it goes through a Bunsen flame, it disappears. Now, that's pretty hot, but a slower reaction is going on all the time. And in our atmosphere, any organic molecules that you formed would be destroyed. So one needed a new idea, a big new idea. And this new idea was put forward 30 or 40 years ago by a Russian called Oparin and an Englishman called J.B.S. Haldane. And they pointed out that the Earth is very old and it's changed a lot. And when it was formed, maybe it didn't have this atmosphere. And there's a lot of evidence for that. There's evidence for that from the rocks, that there was no oxygen at the beginning. This oxygen wasn't there at the beginning. And there's evidence also, of course, from other outside planets, like Saturn and Uranus, which have not this atmosphere, but have a reducing atmosphere. In place of these, they have atmospheres consisting of ammonia and methane and water. These are the molecules, the simple molecules which we started with. And they have no ozone, because the ozone, which we mentioned last time, is made from oxygen. Well, now that's a very different atmosphere, and it's not surprising that it was like that. That is the atmosphere, because when the atmosphere was when the Earth was first formed, it probably lost all its atmosphere because it's so hard. But then the Earth outgassed, and we got this reducing atmosphere, as it's called. And so we look at these molecules and ask how they could have been formed, first of all. The most plentiful element on the Earth, in the universe rather, by far, is hydrogen. And so there's probably a lot of hydrogen around in the first place, and that alone tells us that we can't have had oxygen as well. Oxygen and hydrogen will mix perfectly well. I've got oxygen and hydrogen in this bottle, but they're not very stable. And if we had oxygen and hydrogen in the atmosphere together, they wouldn't get on at all. They would react. And what would they become? Water. And of course, we have water there. So we couldn't have oxygen and a reducing atmosphere. And this is what our atmosphere was probably like when it started. And now you can begin to understand, perhaps, how we can build up our there's an amino acid, like one of those on the Christmas tree. This is the simplest one. It's glycine. And you can see how if we broke up these various simple molecules of methane, ammonia, and water by light, always the light coming in, and ultraviolet light at that, because none of these molecules are colored, remember. So they have to absorb the ultraviolet light. This couldn't happen before because we had the ozone layer. And you remember, ozone absorbs ultraviolet light. I'll show you how it does that later. But once the ozone is removed, once the oxygen is removed, the atmosphere is transparent to ultraviolet light, which can come down, be absorbed by these molecules, and break them all up, and perhaps make little green men? Well, that's a bit ambitious. Perhaps make at least amino acids and nucleic acids, these basic 24 molecules which we want to start with. Well, that's all hypothesis, that's all a suggestion. It was a suggestion until 1953, when one of the most important experiments in this field was done, and it's so simple in idea, in concept and everything, that I'd like to show it to you. It was done by Miller. It was done by Stanley Miller and Harold Urey in California. And they said, well, let's take a primitive atmosphere and put it in a bulb. And here's the water. And if I boil the water it will have a rainstorm in here, and the water will come down here. And just for good measure, let's have a thunderstorm as well, and turn on the lightning. So we pass an electrical discharge through this mixture. And they did this for about 24 hours, and the gases circulated, the rain fell through the lightning, and it went on and on for about 24 hours. And then, to their delight, they looked down here. We can switch off now. We've done it for 24 hours. We look down here, and you see a yellowish liquid. Now, these are quite colorless gases. And a yellowish liquid has collected down here. And they took a little of that liquid out, and they analyzed it. They analyzed it by chromatography, as it's called. This is a matter of letting the various molecules fall down blotting paper and seeing where the spots get to. This would be a mixture, and these would be 3 unknowns, and they found that if those were amino acids, if they put amino acids on, then they got the same spots at the same distance on their chromatographs. That was the simple method of analysis. So they had made amino acids just by passing an electrical discharge through a primitive atmosphere. And I always like to describe that experiment because it's so simple. Any of you, because you're all very, very bright and intelligent people, any of you, I'm sure, could have had the idea of this. Maybe that's how life started. And you could have done the experiment at school. You saw the apparatus. It's not very complicated. You saw the method of analysis, a bit of blotting paper, chromatography, and that's what was done. So, now, then, we have... Let me just say that they used an electrical discharge. They're really good at... What would have been a better experiment? Yes, to use light, because there is 100,000 times more sunlight energy than there is electrical energy, but that's difficult. So very sensibly, they did the easy experiment first. Since people have used light, and they've built up amino acids and the nucleic acids and all the other, the sugars and everything nice, they've built up in this way. Well, now, okay, so now we've got the basic building blocks from which life can be made. And now you might say, yes, but we've a long way to go. What about putting them together as DNA and proteins and so on? And especially since I've told you that things tend to get mixed up, didn't they? You know, the chaos is the natural order of things. Well, if the sun has pumped enough energy into molecules, if the molecules are energetic enough, they can build up. And I'll show you an example of this. Here is a couple of liquids which I'm going to mix and then stir. And what I'm going to try to make is a polyurethane. Polyurethane is a material, quite important material now. It's used for making tires and cushions, and particularly it's used for insulation. And I want to show you that it will form itself spontaneously. It has to be stirred for quite a little time to get the reaction going nicely. And now we've stirred it enough, and if I put it there, you see the reaction is spontaneous, and the volume of the product is far greater than that of the separate parts from which it started. And so the reaction is going on spontaneously. And this is a building up of order from disorder because it's these molecules are much bigger. They're linking up into long threads. And the little bubbles in there, that's what makes it such a good insulator. We'll let that cool down, and then I'll show you what a good hat it makes later. Now that is perhaps not very much like... That's perhaps not very much like... a protein, but nylon is. Nylon is very much like a protein. Nylon was discovered by an American chemist called Dr. Carruthers, who was trying to make silk, and silk is a protein. And he said, I'm going to put together molecules to make a protein-like structure. And he took molecules which were rather, not quite the same as this. There are a number of ways of making nylon. These are the ones I'm going to take to make nylon and to show you this. He took a molecule with 2 amino groups on either end, and this is just a hydrocarbon in the middle. Molecule with 2 chlorine atoms on either end, and a molecule with amino groups. So he took a lot of those and a lot of those and mixed them. And when he mixed them, a reaction occurred. These are combined to form. These combine to form. I pulled the wrong one off, haven't I? I pulled the oxygen off. I'm sorry. These combine to form hydrogen chloride. And so these molecules could link up like this. But you see, they're reactive at both ends. So the other end can do the same thing. And we've got a molecule twice as long. If I do it again, form another hydrogen chloride, and link up again, we have a molecule 3 times as long. And so you can go on and get the long threads of nylon. Let me show you that this actually does happen spontaneously. Here I have, this is the one with the chlorine atoms on, which is called sebasoyl chloride. I'll pour that in first because it's dissolved in carbon tetrachloride, which is heavy. And this is dissolved in water. This is the other one, the hexamethylene diamine, which I'll pour on the top of this first layer, so. And you can probably see already perhaps a bit of hydrogen chloride coming off, which we would expect from what I said. And you can probably also see some goo has been formed. Can you see some goo? Oh, yes. Right. Well, let's just pull out a bit of that goo, like this. See? And pull the goo out, some of it, and a bit more. Now, very shortly, with a bit of luck, if I can get in from the wall, we shall get a thread of nylon. The nylon tends to stick to the wall a little bit. There we go. Thread of nylon. And we can spin some more nylon. We'll put a little spinneret on it, like this. Put this over the top of the beaker. So, do a bit more fishing. Pull the nylon out. There's a thread. Put him over the spinneret. And then I don't have to work very hard if gravity will do it for me. Let's see whether gravity will do it for me. There it goes, and it'll go on spinning until it's used up all the liquid. And that's a nylon thread. And that has been, let's let gravity have a bit more chance by raising it. And that's going spontaneously all the time, you see. The molecules are combining spontaneously without any help, because these molecules have enough energy to do so and to combine spontaneously. Well, now, so we see how you get organic molecules made by the sun. We see how they oh, let's see what's happened to our hat. It's such a good insulator, this. It's full of air. It's such a good insulator that if you bore a hole in there, you can make a very good Panama hat. The next stage is to ask, you may say now, all right, but even these big molecules are a long way from cells, aren't they? Cells have outsides and insides and a lot of structure. Well, it so happens that it's very easy to bring about the next stage. If you just mix protein and sugar in water at the right acidity, you get cells. Not living cells, but they're cells. Fox in America heated some amino acids, made a protein, and then he dissolved them in water, and this is what he found. he found when he looked under the microscope that he had these little cells, you see? Just like you'd think those were living things, wouldn't you? They're not. There's nothing alive about them. And what is more, you see, some of them seem to be linked up. And if we look at another picture, a bit of a blow-up here, this is another picture taken by Fox. You see, here are two cells just joining up. Here is one beginning to divide, and here's one that's divided. They're not alive. It's just a spontaneous process, a colloidal process which occurs. And it's not all that surprising, really, because you've seen bubbles do this, haven't you? I mean, if you blow bubbles, they break up and they come together. And that's all that's happening. We shall see how that happens later. Well, now, so it's all looking very promising, isn't it? We're almost at that little green man. We've built up cells and so on. There are some problems. There's one problem I'd like to tell you about. And that is that these molecules, I'm sorry to have to tell you, these molecules on the Christmas tree, when we make them, we don't make them quite the same as nature does, because nature makes them all left-handed. These molecules, all of them except glycine, can be left-handed or right-handed. Now, that's a strange thing for a molecule to be, isn't it? But it means exactly what I say. If you look at it in a mirror, one is the mirror image of the other. This is methane, which we looked at before. If we make those 4 atoms different, like this, then we can do that in 2 ways. Look at those two sets of balloons up there. Are they the same? Assuming the dots are exactly the same and so on. I put the dots on so that if you're colorblind, you can see what they are. Are those two the same?
No.
Well, we say almost. What's the difference?
Same balloons, but in different order.
Different order. Would you like to come down and tell me more about what you mean? It's true. They are different. I hope. Let me have a look. Green, blue. Yes, they are different. Would you like to come and hold the other one? Now, you see, bring them down a little bit further, will you? There you go. You see, you could not superpose those. If I put the red there and the yellow there, the blue and the green in the wrong order. The way to check, I find it very difficult sometimes to check. The way to check is look along the red, towards the red, and then go round clockwise, and that goes what? Green, yellow, blue. And what does this one go? Green, blue, yellow. And you can't change one into the other. At least, not easily, you can't change one into the other. You can't superpose them. Can you change one into the other, do you think? Have a go. The molecule, if it tries really hard, shall I help you? I think he knows how to do it. Good, very good. He's pushed. Let's see whether he's done it. Now, green, yellow, blue, green, yellow. Oh, we're the wrong way up. Look at the red. Hold him that way so red's upwards, eh? And then we have green, blue, yellow. Green, blue, yellow. You've done it. Very good. Well, you see, it's very hard for the molecule. It's as hard for the molecule. What was your name? What was your name?
Adam.
It's as hard for the molecule to do it as it was for Adam to do it. In fact, the molecule doesn't manage it. The reason we know this is that molecules like this have an interesting property. Remember last time we talked about how light was polarized, didn't we? Like this. Now, if polarized light goes through a molecule which is asymmetric, like this, it's turned a little bit. I can show you how that happens. I have 2 cross Polaroids here, and I'm going to put in front of them 2 sugars, one of which rotates light to the left and one of which rotates it to the right. Now, if I cross the Polaroids, you see how they go dark and light and dark and light. And now if I put the 2 sugars in front, let's see what happens. Put the 2 sugars in front, and now I rotate the Polaroids. You see they behave differently? And they go through all the colors of the rainbow. but in a different order. In fact, in complementary order. Brown on the right, blue, then pale blue on the left, and so on. And of course, you get the colors because different wavelengths of light are rotated by different amounts. I can show you the same effect rather a pretty way. It doesn't add much to what I've been saying, but it's rather a nice way. And it's one way we might contrive to do at home because you only need one Polaroid. You can use one. You don't have to break your spectacles to do this one. You have to do that. You need one Polaroid and you can just do that. If you fill a tube with sugar, pass light up it, but put the light through a Polaroid first, let's have a look at what you see. Lights down, please. Let's have a look at what you see. Now you see, not only do you see the colors, you see how they're spiraling upwards. Bill Coates is turning the Polaroid, you see, as we do this, and they're spiraling upwards. Now, the interesting thing about this, I want to just hold it there, Bill, that'll do. Now I put a bit of black tape round there so you can all see it, and I want those in front of you to tell me what color is where the black tape is.
Green.
And those on my right?
Red.
And those on my left?
Red.
Well, somebody's colorblind. Isn't that ridiculous? Isn't that ridiculous? Let me put it somewhere else. Put it somewhere else. Now, what do you in front say?
Red.
And what do you on the right say?
Green.
You on the left?
Green.
Well, I don't know about that. Right, lights. I just hold it up. Very quickly, I don't want to go into this too much, but very quickly, you're seeing that light because it's scattered. Scattered light is polarized, as you can check by looking at the blue sky through a Polaroid. And light in that direction, over there, if it's coming up like this, would have to be polarized like that. And light towards you would have to be polarized like that. So they have opposite polarizations, and you see different colors. All right. Well, now, so that was a bit of a problem. If living molecules have to be left or right-handed, how does nature cope with this? And we don't really know the answer. Some people say because the sky scatters polarized light, that might have an effect. It's more probable, really, that both left-handed molecules and right-handed molecules, those 20 molecules, 19 of them anyway on the Christmas tree, were formed both in left-handed and right-handed forms. Two sets of life evolved, but at a very early stage, one won out. They had a battle, and one won out. One evolved in some way an advantage which enabled it to win out over the other. So that's the situation there. Now, let us see where we've got to on our clock. We have then been going through this area before life, and we have got to a stage where we think we can see how life might have just been formed. We've certainly got to the big molecules, and life could have been formed. And so through this doubtful gray area, we're now in this area, white. Now, I've painted this differently. I painted this white and not green because in this area there was no green. We were not in those days absorbing light, visible light. We were living on this mass of molecules which had been formed in the dilute soup of the oceans, the sugars, and so forth. And the energy that living things in those days was got by fermentation. Where's glucose? Hold up your glucose. Now that is a good food. And when that ferments, it breaks down into 2 molecules of alcohol and 2 molecules of carbon dioxide. Alcohol is a good food as well. I mean, I really mean that. You make, in moderation, you make alcohol all the time, whether you're teetotal or not. And you get much more energy by breaking that down as well. Okay, thank you. But what, in this white area here, it is fermentation. that life is living on. Life is living on the process of breaking down sugar into alcohol, and of course it does this very usefully for us in this particular case. I shall have to get into practice before Christmas. It does this very usefully for us. I'd like a mop. Thank you. Join me later, will you, in it? Thank you. And I can taste the alcohol. You saw the carbon dioxide. And what is more, you saw the energy. The energy of that fermentation reaction was liberated in part by the energy at which it expelled the cork. And so, we can see how this part, how energy was derived in this part of the process. But now things begin to, a crisis arose in the history of life on our atmosphere. Because all the time the sun had been breaking down these molecules, as we have seen, breaking down ammonia into nitrogen and hydrogen, breaking down methane into carbon dioxide and hydrogen, and breaking down water into oxygen and hydrogen. And so we had we're getting very near to our present atmosphere. But of course the thing would go backwards as well. The hydrogen breaking down and reforming the original molecules. But there is one difference. Hydrogen is very light. If you fill a balloon with hydrogen, it goes up, doesn't it? And it'll go up and up and up and leave the atmosphere altogether. And that's what happened. So gradually we lost the hydrogen and we got our present-day atmosphere. And another thing happened. Not only did we form the oxygen, but oxygen molecules, the O2 molecules, when broken up, form O3. And they form this all-important ozone layer up here, right up at 15 miles high. Very, very thin layer, but all-important because it stops the ultraviolet light getting through to us. Now that's very important. If the ultraviolet light weren't stopped, life couldn't crawl out of the sea. It would be burned to death. Let me show you. Have we still got those little animals under our microscope? They're there? Well, if we've got those under our microscope, Mr. Coates is going to shine ultraviolet light from this lamp on them. Can we see them alive first? Can you see them all running around? Are they alive? Now shine ultraviolet light on them. Just above the microscope slide. There's no cover slip because glass would absorb it. And there he's shining it on. They're looking a little bit sleepy and they're dead. Some are drifting across, that's just the flow of the water. I think not a single one is wagging its tail. Isn't that a cruel experiment? Lights on, please. And that's what would happen to you. That's what would happen to you if you've got too much ultraviolet light. It would certainly kill all the leaves and so forth. So we're very fortunate not to have ultraviolet light. A dangerous thing to have on the skin. It can cause cancers and so on, and this is why people are so concerned at the moment about preserving our ozone layer, because if we lose it, worse than cancer on the skin, we would die altogether because all our vegetation would disappear. So we have seen now how the atmosphere changed round about this time. Around about 2.5 billion years ago, the oxygen appeared, the ozone layer, all chance of further synthesis by the sunbeams directly was removed, and a crisis occurred, and life had to do something very quickly. It had to synthesize a molecule which could absorb visible light, and it synthesized chlorophyll. The green of chlorophyll. The green of chlorophyll and plants took over the photochemical stage, and next time we shall talk about the green area and the area of the leaf. Before we do so, though, perhaps we should... I kept talking about little green men, and it's a bit disappointing not to make a little green man, so maybe one of you would like to make a little green man, would you? Shall we try and make a little green man to finish with? Are you going to do it? Good. Okay. Well, would you like to hold that? Now, here is a green liquid. Hold it absolutely still, because I'm going to pour this onto it. Can you hold it level? Absolutely level. Like that. Thank you. Now, will you look carefully and see whether you can see a little green man there? Can you? Can you see any little green men?
No.
You can't? No little green men? Well, what's missing? What do we have to do? After all I've said, what do we know is essential to form living things? Sunbeams, light. Right. So let us put it on there and shine a light on it and see what happens. And if you watch that, can you see? Of course, I have to have some DNA, some template underneath. Can you see anything happening to him? I think molecules, big molecules are being formed, aren't they? I think they're being formed. Let's have a look now. Let's just take him off and pour the excess liquid into here. And there we have a little green man. You made it. Here you are.